All right, guys, we are moving on to the last section of Intro to Animals. So we're going to learn a little bit about how animals are fertilized and develop throughout time. So, first, fertilization. The male of a species creates sperm in its testicles, and the female of a species creates eggs in their ovaries. And these things come together after copulation, or sometimes copulation isn't necessary depending on the animal, and creates a zygote. So the gametes are the egg and sperm. The egg are very large in comparison to the sperm. They have a lot of yolk in them because the baby actually will just live on that yolk depending on the type of animal. If it's an egg-laying animal, they're going to live on that yolk until they are born. In the case of a placental mammal, they are going to have a little bit of yolk to last until they have implanted into the placenta and implanted into the uterus and created their own placenta. So different animals have different things, but they all have a yolk depending on how long that food supply needs to last. And then there's the sperm, and the sperm have a small head and a long whip-like flagella. They don't have a lot of other things. All they really have in there is the DNA they need to get the DNA into the egg, because remember, you are half mom, half dad. All animals are half mom, half dad, unless they are sexually reproduced, which is very rare. So you get that because you have half of mom's information from her egg and half of dad's information from his sperm. Now, I don't know, even on this picture you can see that this head of this sperm is very clear right here at this tip. That's because it has something called an acrosome, and that's inside that acrosome. This tip right here is a little bit of an enzyme, and so when it crashes into an egg, that enzyme actually helps open up so the DNA can get from the sperm into the egg. And as soon as that happens, actually, it's pretty interesting. Electrical changes happen to where a second sperm or a third sperm can't come in and add their DNA, because that's too much DNA, and that's going to cause a major mutation and probably actually not a real viable pregnancy or birth, offspring. All right, so the sperm cell fuses with the egg membrane. That acrosome will burst and create a hole where then the DNA of the sperm can enter and fuse with the nucleus of the egg. And again, that fusion of the egg causes electrical changes that blocks the entry of any other sperm. So fertilization happens when the sperm and egg get together. The sooner sperm and egg get together, you are a zygote. You are officially have the right amount of DNA for the species that you are. So half of the species comes from mom and half of the species come from dad. I know I just pointed the wrong thing. Mom, dad, there we go. For humans, 23 is in the egg, 23 chromosomes are in the sperm. For a total for humans, 46. Now that number changes depending on the insect or the animal. Fruit flies have 8 total chromosomes. I think dogs are 72. Don't quote me. It's higher. Anyway, there's quite a bit. After you have fertilization, now it needs to replicate to make it a multicellular organism, because right now it's an organism with just one cell. So first you get fertilization. You get the zygote. Then you get cleavage, and cleavage is a separation. I mean, we know we use it to describe breast, but that's really, you're just looking at the separation between two separate ones. So cleavage means separation. So you have your single cell and it divides in half, making 2 half identical cells. Then it divides again and makes a 4-stage cell, and then 8, then 16. Eventually you're going to get a blastula. A blastula is between 16 and 64 cells, and it is a raspberry-esque shape. It's hollow in the center. So all these cells have come together, and they have that hollow center. So that's a blastula. Then it has to go through the process of gastrulation. Gastrulation is the process of making this cup. So I consider gastrulation like a poking. and it's not. Nobody goes up and pokes it. But so you have your blastula, your single circle, a hollow circle, and then it starts enfolding on itself, creating this cup. You have an opening that goes in. So, and that once that has been formed, it is called a gastrula. The whole thing is a gastrula. The actual inner portion is the archientron, and so that's this little cup on the center. And it's going to become your gut for anything that's going to be a vertebrate. Anything smaller than that, it's different necessarily. But so this is going to be, this first opening is going to be your mouth or your anus. And I'm saying you as general animals, not humans. So it'll be the mouth or the anus, and then this is where the food will pass through. And eventually, I'm going to go back to the other ones, it will create a hole on the other end too, making the entire gut. So food goes in the mouth and through the gut, out the anus. Now, I talked about germ layers earlier in an earlier video. Remember, you have ectoderm, endoderm, and mesoderm. The ectoderm is going to be the blue on the outside. The endoderm is going to be the yellow on the inside. And the mesoderm is going to be the middle and form in the center. Right now, in this picture, there's no mesoderm. Right now, in this picture, it is stopped at 2 tissues. But that's just because the mesoderm hasn't fully formed. It's coming. So, there are different patterns of development, how these things separate. A protostome is anything whose mouth forms from that poking, from gastrulation. So once you have your circle, the first hole that is created, if it's the mouth, it's a protostome. Protostomes are going to be mollusks, annelids, and arthropods. So this first hole becomes the mouth. Also, all protostomes go through radial cleavage. So what that means is it looks like a brick pattern. So it splits and twists, splits and twists. So the line of one layer goes in between a cell of another, again, kind of like a brick pattern. So that's spiral cleavage. And protostome, mouth form first. And the other thing is schizocele. I know this is a weird word, schizocele. Schizocele is going to be when the mesoderm forms, this red, between, here at the tip next to the mouth, between the endoderm and ectoderm. There we go. So once it's forming there, and it'll grow all the way out until it looks like this. By the fate of this, it's actually pretty darn similar on the other one, but it's just how it gets there. So protostomes have spiral cleavage, schizocele, and the mouth is formed first. Deuterostomes are when the first hole, the blastopore, this first hole, becomes the anus first. This is going to be echinoderms and chordates. In other words, you are a deuterostome. And I do remember this because I'm a child, and I say deuterostome, doo-doo, he, anus, doo-doo, ha, ha, ha. Yeah. So that's how I remember it. My anus is formed first. So actually, technically, you can even say your anus is the oldest part of your body. So the anus becomes first, and then the mouth is formed second. It undergoes radial cleavage. So what that means is that it splits, and there's no twisting. So all the lines line right up together, like tile. Instead of brick, where they are offset, this is like tile, where they all line up. Then there is enterocele. Enterocele is when the mesoderm forms at the bottom of the cup, or top of the archientron, however you want to say it. Here, away from where the ectoderm and endoderm get together, over here in the center. And then it grows, and then the mouth forms second. So in deuterostomes, we have radial cleavage, everything lining up. We have enterocele, the mesoderm forming at the bottom of the cup, and the anus forms first, and then the mouth forms. So that is deuterostomes, and we are deuterostomes. So there's some hard terms, and they sound crazy, but they're not too hard to understand. You just have to remember their crazy names. So here is a versus again, protostomes spiral, deuterostomes radial cleavage. Protostomes schizocele, right here where the ecto and endoderms meet, and enterocele at the fold of the archaeantron there. And then in blastopores, mouth is first, and protostomes, mouth is first, deuterostomes, anus is first. So here is the actual definitions of what I've already told you. Schizocele is when the mesoderm forms that splits away from the interior of the gastrula. And enterocele is when the mesoderm forms at the top of the archaeantron. So again, where they come together, ectoderm and endoderm come together for schizocele, and at the top of the archaeantron cup for enterocele. Entero being inside, kind of. All right. Remember, for the body cavities, we have coelomate, acoelomate, and pseudocoelomate. We went over this last time. So you have, remember, a coelomate is when the mesoderm fully lines this, and that's going to be anything that is not a kind of a rotifer or a roundworm or a flatworm. A pseudocavity, a pseudocoelom, has the mesoderm on the outside but not the inside, and it has a fluid-filled cavity. Those are going to be your roundworms and rotifers. And then your flatworms are going to be acoelomates. They have no body cavity. And that's it for the first chapter. That's the basic gist of how animals develop and a basic gist about animals. So take a second to make sure you remember what protostome versus deuterostome means, what radial versus spiral cleavage means, and what schizocele versus enterocele means. And remember the different types of coeloms, the coelomate, acoelomate, and pseudocoelomate. Don't forget where the sperm and egg come from, the gametes, and don't forget when they come together, they make a zygote. All right, that's it for Intro to Animals. We are done with this chapter. We will have a test soon. I will talk to you guys later.